I'm an RNA scientist, and during the pandemic, I've been spending part of my time in public education, giving talks, trying to relay the excitement about some of the recent breakthroughs in RNA science to the general public, because I find that people understand the DNA double helix pretty well. 23andMe, genealogy, forensic medicine, but RNA leaves them baffled. And although scientifically there's not much difference. So I usually start with a little joke, but there's some truth to this, because the main difference between RNA and DNA is in fact the single-stranded nature. They have the same informational content. And so if there's an AGC along one strand of the DNA, there will be an AGC in the RNA copy of that DNA. I also try to give a little local character to this because of the recent Nobel Prize. And if I'm giving talks in Colorado, it's fun to talk about Jennifer Doudna, who did her postdoctoral training in my laboratory in Boulder. So how does this CRISPR genome editing work? Well, it's not as simple as the red scissors cutting the DNA. What actually happens is that RNA, a guide RNA, searches along the DNA double helix for a match. And when that match occurs, a protein component called the Cas9 enzyme cuts both strands of the DNA at that point. And if you want to cut a different gene in the human genome, you simply engineer in, and my undergraduates in my lab do this routinely. You put in a different guide RNA and you cut a different part of the DNA that matches that. There are a lot of exciting research applications. This is all within the last 10 years, okay? But now we can surgically precisely edit the human genome to inactivate or edit a genetic mutation in a gene to test the function of those. But what I'm going to talk about in the next 2 minutes is just how we can add a little fluorescent light bulb to a protein using CRISPR genome editing to follow the protein as it explores a living human cell. So the particular system that we study in my lab is telomerase, which is a molecular machine that extends the ends of our chromosomes. Liz Blackburn, Carol Greider got the Nobel Prize for discovering this enzyme. And this is medically very important, because if you have too little of this enzyme, you can't maintain your stem cells, but too much is one foot in the door for cancer, because this is the immortality enzyme. It is by extending telomeres, it allows limitless proliferation of human cells. So what Jan Schmidt, a postdoctoral fellow in my lab, did was to use CRISPR genome editing to put sequences encoding a fluorescent tag in front of the telomerase protein, and then a different color in front of a protein that binds to the telomere, the end of the chromosome. And now this is just mind-blowing technology. When I was a graduate student, if someone had said this was possible, I would have said, you're crazy. But we can follow single molecules in real time moving around a living human cell. So the green molecules are the telomerase, the red are the chromosome ends that they are searching for. And this is not sped up. This is actually the dynamics within a nucleus of a human cell. And of course, when you see the co-localization of the green and the red, that means that the telomerase has found its target. Now, the other key area that RNA has been in the news for is, of course, the mRNA vaccines. A vaccine needs to present a part of the virus that will be observed by the immune system. It sort of primes it, says, if you ever see this again, this is trouble, do something about it. And for the coronavirus, this is the spike protein. Well, what clever scientists at Moderna and at Pfizer-BioNTech found is that instead of using the whole information transfer in biology of DNA makes messenger RNA makes spike protein, you can just short circuit this, and you can make a stabilized messenger RNA and inject that into arms, and that will instruct the body to make the spike protein, which is then the immunogen which serves as the vaccine. Now, how is it possible, given that 0 messenger RNA vaccines had ever been approved, had ever been successful, how could the first one be successful? And this is why this is a great public education story, because it's really the taxpayers' dollars that through the National Institutes of Health and other funding agencies led to a lot of fundamental research, which was not disease directed, but taught us, for example, how to make loads of RNA in the test tubes, how to make it stable and safe. And those were key then to construction of the coronavirus vaccine. Thank you.